and welcome to today's webinar hosted by Solaris Bio. We have a very exciting event today. We'll learn many interesting things, including the discovery path of novel human DHFR inhibitors, mechanism of action to circumvent cell resistance, and also cell target engagement for hit validation and optimization. Today's webinar will be about 30 minutes of presentation, and then after that, we'll have opportunity for some Q&A. So if you have any questions or comments that come up during the course of today's event, please put them either in the chat or the Q&A, and we'll attend to all of those questions and comments at the end. Today, we have the privilege of hearing from two excellent speakers. I'll give you a heads up of who they are before their presentations. First of all, we have Dr. Dennis Wright. Dr. Wright holds a bachelor's degree in chemistry from West Liberty State College and a doctoral degree in chemistry from Ohio University. Dr. Wright is currently professor of medicinal chemistry at University of Connecticut and chief strategy officer at Quercus Design Group. One of his research areas includes therapeutic development for infectious disease, neurodegenerative disorders, and cancer. Also, Dr. Wright has recently improved upon a new way to thwart drug-resistant tuberculosis. So Dr. Wright, thank you so much for all that you do and also for being here with us today. After that, we're going to hear from our co-founder here at Solaris Bio, Dr. Ivan Babik. Dr. Babik is an accomplished cancer researcher with a PhD in biochemistry and molecular biology from the University of Calgary. He has over 15 years of experience in drug discovery and has been actively involved in preclinical and clinical drug development. Dr. Babik holds patents for small molecule therapeutics and is inventor of several novel drug screening technologies. So we're looking forward to hearing from you as well. Okay, so thank you so much for everyone for being here on time, for hearing us, and we're going to turn it over to Dr. Wright to hear what you have to say. Take it away, Dr. Wright.
Oh, okay, Emily, thank you very much for the nice introduction, and thanks those of you online for coming here today. And I'm going to tell you a little bit about a compound we've been developing for the last several years. And it's going to be an inhibitor of an enzyme known as dihydrofolate reductase. I'm talking about some of the unique sort of sides of these types of molecules, how they differ from some of the currently used DHFR inhibitors, and sort of forward-looking where we're trying to sort of look at these in a more translational sort of manifold. So we'll sort of go ahead and so this is presentation. That's obviously my title. And then also the other person who's been a big contributor to this is a good longtime collaborator, Charlie Giardina, who's also at the University of Connecticut in the molecular and cellular biology program. And he does a lot of the nice cancer biology work that I'll show you that sort of underpins a lot of what we're doing on the drug structure-based drug design front. So the target we're looking at is a target that's been a pretty well-recognized anti-proliferative target in both infectious disease and oncology, as well as inflammatory disease. And this sort of large path kind of shows this very important sort of pathway. And this is really the whole sort of root of one carbon metabolism in cells. So all the methyl cycle and sort of one carbon donors work through this pathway. It's a pathway that is conserved in almost all domains of life, from simple prokaryotes to eukaryotes. And this is the way that one carbon donors are created as sort of the production of really important building blocks like thymidine and a variety of amino acids. And this is an area my lab's worked on for about 15 years now, using primarily high-resolution crystal structures to design compounds specifically against drug-resistant versions of some of these enzymes, and a little bit about how we sort of ended up with the design driven by that structural analysis, but it has a lot of other really interesting features on it. So like I said, it's a pathway that is present in both prokaryotes and eukaryotes. And is that visible? Or let's say I'll annotate my screen here. Let me try the annotation. And then the green one here in this side, this is a pathway that only really happens in lower organisms, primarily in prokaryotes and things like this, where they will do full de novo synthesis of a lot of these precursors, and these are related to B vitamins and really important sources of the enzyme folic acid. But we don't do a lot of the early steps in this, and instead what we do is we rely on dietary sources for a lot of our folic acid. So we know it's a really important supplement for pregnant women and for development, and it's really valuable, specifically when there's a lot of proliferation going on. And the heart of that enzyme, the heart of this pathway is an enzyme shown here, does two sequential steps, and that's an enzyme known as dihydrofolate reductase, or DHFR. And so what it does is it takes folic acid we get dietarily, it reduces it originally to dihydrofolate, so it adds a hydrogen there, and then it can add a second one in to make dihydrofolate. So this is an NADPH dependent enzyme, and it's a very fast moving, high turnover enzyme that is involved in recycling these factors. So what happens with sort of these folate derivatives, though, is that once you make tetrahydrofolic acid, this will pick up a carbon atom shown here from serine to make methylene, methine, or formal or methyl donors. So every one carbon building block that comes through is made this way. So we think of a lot of really important steps with this. One of the most important is shown here in the lower left quadrant. And that is the production of deoxythymidine monophosphate from deoxyuridine monophosphate. So this is the base T. So you really can't make DNA without this enzyme. But also there's a lot of other maybe less well-described pathways that are shown over here, for example, where we make methyl tetrahydrofolate. This is important in turning over S-adenosyl homocysteine to make SAM donors. As well as some of the things involved in purine biogenesis. So we have all of these really key building blocks needed for cellular proliferation, and all of them hinge on the ability of this reductase to turn the oxidized cofactors back over after they've been depleted. And so this has been a pretty well-recognized target, and we've seen a lot of application of this in the antibacterial world, for example, with trimethoprim. In malarial field has a lot of examples of these, and in the cancer field, specifically the compound methotrexate, which has been used since about the 1950s, primarily in the treatment of pediatric leukemias, as well as some other diseases. So it's really been a very important sort of area, but there is a lot of drug resistance that shows up with this. So there's drug resistance both in all of these pathogens as well as in the cancer cells. A lot of that resistance was based on mutations to the dihydrofolate reductase active site. And that's really got us interested in that. I think as we've gone on, we've learned a lot of the resistance, though, is more complex than that. And I'll show you some things that are important about, for example, cell transport and things that also become really critical mechanism. But we started out sort of looking in that kind of vein. And so let's see here. And then see if I'm advancing here. Okay. Oh, somehow I kept my annotations. Not sure. How do I erase those, Emily? That was in
Either the
the
little trash can or the erase button on your little panel.
little trash can. All right. Let's see if I can find said trash can.
About 2/3 of the way down. I'll take care of them. There you go.
Okay, that would be great. I
Yeah.
have to be
No, no
cautious
problem.
doing.
No problem.
See if I can back up here. So, okay, so the all right. So the design that we sort of that sort of preoccupied a lot of our time was really designed against these sort of resistant enzymes. And this is a group of compounds we refer to as propargyl-linked antifolates. We call these PLAs for short. And the sort of generic design is shown here, sort of at the top of this. And what it has in it is a couple different domains that are really important. And kind of really what was critical to this, although it looks kind of small in the drawing, was the use of an acetylynic linker between a diaminopyrimidine head group and this sort of large hydrophobic domain. And the reason we really keyed on that is that when we looked at structures of many of the resistant enzymes, they had mutated residues near that domain into very large hydrophobic residues like phenylalanine and isoleucine, and it kind of got in the way of a lot of the inhibitors. And so the idea with the acetylene is the SP hybridization of that gives you a sort of a long linear sort of motif, and that allows it to slide through really what's a very tight channel in a lot of these enzymes. And that's sort of been the heart of it. It also makes it a pretty easy way to make a lot of derivatives because it allows a sort of modular synthesis where we can hook sort of that diaminopyrimidine head group together with these hydrophobic domains. And that works pretty well and sort of allows us to make a lot of compounds quickly. And so we've had a lot of projects in this, both in the infectious disease space, but also looking at oncology. And that's what I'm going to talk to you largely about today is a candidate that we have primarily looking at in AML leukemias. And so sort of just to compare a little bit about the structure and talk about why these are different. So the top compound here is methotrexate. So again, this is a pretty old compound. It's a very, very close analog of the natural folate cofactor. So it really mimics that structure quite a bit. It only has a very small change in that diaminoquinazoline sort of portion of the molecule in blue. And it's linked, and so you have methotrexate. There's an aromatic core in the middle. This is an amino acid called paraminobenzoic acid. That this is what sulfa drugs inhibit the synthesis of. Again, we don't need to make it because we get that from our diet. And then the MTX derivatives have, let me see if I can get back to my annotation here. They have this carboxylate, sort of this glutamate tail on them. And that's a really important feature of these. Again, those two carboxylic acids are found in the natural folate cofactor. They're really important for creating interactions in the active site, but they also impart a very high degree of negative charge. So at physiological pH, this carries a -2 charge. And so it gives it a very low log D of around -4.6. The compound that we've come up with and sort of developed in our time is this compound known as UCP 1162, or just 1162 for short. And this is what it has a somewhat different structure. So you notice there is some similarity in the blue region where we have these diaminoaminoperimidine head groups. And these are really important for interacting strongly with a highly conserved active site, aspartic acid or glutamic acid. It also shares a hydrophobic central domain in that green ring, but instead of having the glutamate tail, we actually have a second aromatic moiety on there, in this case, a methylpyridine group. So this really changes the physiological properties of this. So instead of having that very negative log D at -4.6, this is a much more hydrophobic compound, and the log D is about 3.9. So there's a real big change in the physicochemical properties of that. And that's going to have some really interesting ramifications for how we think the cells respond to this and where some of the efficacy comes from. So you can see sort of the profile of the two compounds shown here. There's the, in terms of the enzyme activity, where we can look at, methotrexate's about a 53 nanomolar inhibitor. And we actually came up with a compound that was quite a bit better. And we were kind of surprised we were able to do so well. So it was about a 27 nanomolar inhibitor. And that's actually been pretty remarkable because most of the time when you get away from these glutamate units, that really reduces interactions with the active site of the enzyme because there's a series of very positively charged residues, a couple arginines sitting at the base of the active site that anchor that in. And so one of the things people sort of thought about a lot is that without that interaction, you can't get the level of potency out of a compound like methotrexate. But at least by this metric, we were able to get something really potent and very exciting in terms of how we could sort of move ahead with that. And if we look here, this just shows. Thank you, Emily. Awesome. So, but just to give you a little bit of data, we were looking at this compound, and here's sort of the activity of this comparing the activity against 1162 with methotrexate, and that's what we kind of use as our best comparator. But what you can see is that in different lines, colon cancer line, ALL, and AML lines, we had activity that was usually better than methotrexate, again, looking at anti-proliferation. And you can see down here a couple curves. It was done by a student in my lab, Ditum. You can see that we get about a 10-fold increase against leukemia lines. And so we were really excited to see this because in general, people have not been able to move away from that, what we'll call classical antifolate structure. I'll show you in the next slide coming up, and how that compares. And you just really haven't been able to get that kind of potency out of them. And so that was a really kind of a remarkable finding. And I'll tell you why we think we get that sort of high level of potency. Let's see, you can see we were able to show, for example, that the activity of the compound could be reversed with leucovorin. So this is pretty typical. So one of the things, for example, when patients are on DHFR inhibitors, they're given leucovorin rescue to help sort of mitigate some of the side effects of that, largely leukopenias. But it reverses the activity. You can see, for example, both methotrexate and 1162 the anti-proliferative activity is fully reversed by high doses of leucovorin. So that was a good idea that we were on target in terms of inhibiting DHFR, at least from a pharmacological perspective. And so that jived with what we had sort of had seen in some of that sort of preliminary ones. So looking now, getting a little deeper into why we think this is different is that a lot of it goes back to, and we'll see this come up, have a couple ramifications, but really this difference in the physiological properties of the two DHFR inhibitors is really important when it comes to the cellular activity and to cellular resistance. And so, for example, you can see here just sort of showing on this sort of schematic we made the difference between these two in terms of transport, retention, and elimination phenomenon. And so the long and short of it is that the pathway in for these compounds is quite different, and what happens to them after they're inside is very different. So with methotrexate, which is shown here, remember, this is this very, very negatively charged, very hydrophilic compound. So that compound of itself can't passively diffuse across the cell membrane. And so the way that that gets into cells is by the use of active transporters. And these are known as folate carriers. There's several different ones, the reduced folate carriers or FR alpha. And this is the way the cells normally salvage vitamin B out of the B vitamins out of the diet. So it's the way that they get folate naturally. Methotrexate is a really, really close analog of that. So it rides in on the same transporters. But it needs to do this because of the high negative charge. So the route into the cell is different. Whereas if we look at a compound like 1162, for example, which has a much more hydrophobic characteristic, this compound, we believe, passively diffuses through the lipid bilayer. So its transport and uptake into the cell is independent of these transporters. Another thing that can happen in this is once it is in the cell, an enzyme in the cell starts to work on the folate cofactors, and this is an enzyme known as FPGS, or folate polyglutamyl synthase. And what it does is it actually takes that glutamate that's already negatively charged and ligates additional glutamates on. And what you end up doing is building up compounds with a -6, -8 negative charge. And the reason that's done is to enhance cellular retention. So this happens again naturally with folate, but because MTX is so close, it really follows the same path. Inside the cell, then this acts as a DHFR inhibitor. It blocks the activity and it blocks the replenishment of the 1-carbon donor pools. And obviously that's going to block proliferation. So we can't make thymidine, we can't make amino acids, we can't make purines, and the cell simply can't divide. And you see, again, a lot of cell-specific activity. These tend to rest the cells in S phase when you have mostly DNA synthesis going on. So it's pretty consistent with that type of model. So again, 1162 doesn't have that glutamate. And so again, we're not expecting this to be polyglutamylated. Now, so we have two things that happen with folate that are really interesting. We have an active transporter that brings it in, and then we have this polyglutamylation that helps retain it, prevents it from being pumped out by MDR pumps. So methotrexate takes advantage of these sort of natural components of this. And so what happens in resistance is these become Achilles heels for the drug. So what happens in the cells is they can break these mechanisms. So we see lots of mutations of the transporters, loss of function mutation in FPGS, upregulation of transporters, and other things that reduce the efficacy of methotrexate. And the idea is if our compound is independent of those activation and retention strategies, then maybe they'll be more resilient towards those type of resistance mechanisms. And so as we go on, I'll show you a little more data on some of the biology of these. And we can see here, for example, just showing the IC50. And this really got us thinking. So one of the questions we started asking ourselves is, why was this so good? And why did we have so much potency against this? The enzyme numbers were not that different, but we were getting about an order of magnitude difference between the two. And we started, again, if we looked at IC50s, which are kind of a reasonable way to look at enzyme inhibitors, they weren't that different, and you can sort of see these. But what we discovered, and this is an area we've been actively interested in a lot of sort of thinking about anti-proliferative strategies, is the idea of enzyme residence time. And so and this is something we've looked at a lot with different DHFR inhibitors, both in antimicrobial applications and in cancer, which is how long does the compound stay bound after it binds? And this is and you can think of this kind of as the reciprocal of off rate, but it really gives us what's known as the residence time, how long the compound sort of can sit on there. And the way we measure that is shown down here in the bottom, and this is a jump dilution assay. And the way we do this is we have, for example, in the control, this is just the enzyme by itself. We incubate the enzyme with the inhibitor overnight, so it's got a very, very long incubation time. And then that is put into a very, very high dilution of substrate, and we look at how long it takes the enzyme to recover. So if it's bound really tightly to a long binder, long duration binder, then we see a lag time for enzyme recovery. And you can see that both methotrexate do that, and that was kind of, we anticipated that would be true because of all that strong electrostatic binding. But what we were really interested in was 1162 was just as good, in fact, a little bit better at that. And that's something we didn't expect because we'd taken away so many of the interactions that we knew drove a lot of enzyme binding. But 1162 turned out to be a very long residence time inhibitor. And we think that's a real key part of antiproliferation, because as the cofactors are turning over, you start to accumulate substrate. So basically they start to get outcompeted as a function of time. So if you have a very long residence time inhibitor, it's harder for the cell to recover and sort of move that kind of through the sort of shunt in the pathway. We've been working on getting a crystal structure. So we haven't been able to get a crystal structure of this compound for reasons we don't quite understand. We do have some others. This is a docking complex. And what we think we see in the docking, so we need to get a real structure, but this is a pretty good, we're pretty good with docking these, is that we actually get an extra an interaction that we've never seen before, which is shown here, and it's an interaction between the drug and the NADPH cofactor itself in terms of a hydrogen bond. And we think this is stabilizing the ternary complex to a very large degree relative to, so in other words, we've lost some of those key electrostatic contacts with the acids, but we've built in something new that keeps the compound on there for quite a long time. Seems like I'm gonna move along here, so we're getting closer to our time, but we have some knowledge about why we think that these bind so strongly. I'll show you some more biological data. Again, this is just showing the activity is very high, sub 1 nanomolar activity against a number of lines. And the activity is very, very good against, particularly against AML. And so that's a very tough disease to treat when we think there's a lot of opportunity. We've shown, for example, we can see the compounds knock down sort of methyl cycle components, again, sort of validating pharmacologically that we're on target. Some other nice data here. This was really exciting data that we just submitted for publication, where we looked at raising resistant mutants. So this is looking at a parental leukemia line, CCRF cells. And you can see here on the left that over here, that if we treat with methotrexate, you can see initially there's sensitivity, but over time that goes away. And as you leave it on longer and longer, we start to get these very resistant lines, and they grow like wild type. So we get very quick resistance to methotrexate, and again, through mechanisms that are known clinically, usually having to do with impaired transport and retention. What was really intriguing is when we grow resistance to 1162, we really have a very hard time getting it. We can isolate some small resistant clones, but nicely, if we look over here, this is against the MTX resistant lines, as we maintain activity against the methotrexate resistant cell line. So the mechanisms that are being used by the cell to evolve methotrexate in sensitivity are not sensitive. UCP 1162 are not sensitive to those, and so it maintains sort of a robust response in that. And that's really very exciting in terms of looking at the potential sort of evolution of resistance that kind of invariably happens in chemotherapy, but we see that could be a bit slower. We've done a lot of nice work. This is Charlie has done a lot of nice work doing single cell RNA-seq experiments. And so we've really looked at this, and there's a lot of data here, but to make it really long and short, what we find is that methotrexate selects out previously existing resistant cells in the population. So we can find that subpopulation of resistant cells. They're there before treatment, and then they get selected out clonally and expand over time. What we find with 1162 is the population that does eventually rise from resistance is not there at the beginning, and they really have lots of strong markers for stem cells. And you can see that kind of here, that there's a lot of that. So basically what it seems like happens with 1162 is it goes more into a cancer stem cell-like state rather than finding an easy sort of mechanism to resistance. We've done some animal work with this, just real quickly. We've done some inflammatory models. Methotrexate has a lot of use also in treating IBD and RA. We've been able to show that we can see knockdown of inflammatory cytokines in mice. We've been able to look at this in some preliminary, this is our work that's mainly ongoing now, doing AML models, where we've been able to show activity in vivo against an engrafted line in immunocompromised mice, and we can see pretty interesting response to that. And so we're kind of excited. This was done at a fairly low dose because we didn't know how to formulate it. We've come up with it. We paid someone to come up with a better formulation for us. And so now we're repeating a lot of these at a higher dose. And then the last part I'll leave you with just is really interesting is all the things I showed you on 1162 have to do with the racemic compound. And it has only a single stereogenic center where that methyl group comes off the propargylic center. And so we've always done most of our work with racemates. And then recently we were able to do a chiral separation of that. And we wanted to know whether there were real differences between the two sort of enantiomers, right? And sort of thinking about this. And this is just sort of late breaking news we just got, but this is really cool. And you can see we did a jump dilution experiment again. Here's the control. So this is the enzyme recovering on its own. And what we see is that the two enantiomers have very large differences in resonance time, about 20-fold. So one of them stays bound a lot longer. And that's very consistent with our modeling where one conformation can reach that. And then what's really exciting to us is we recently just looked at the two independent enantiomers against, this is MB411 cells, it's an AML cell, and we see at least an order of magnitude difference in activity. So we see it now a very, very strong correlation between this parameter of enzyme residence time and anti-proliferation. And we think that's pretty exciting because that's a parameter we can continue to optimize for looking for how long the inhibitor drug complex stays together. So that's kind of sort of been more the some really exciting news about how the stereochemistry really impacts that. The end I just kind of, whoops, somehow I've lost my, all right. Just thank some of the people involved. I think a lot of them were named on the slides, and NIH has funded this. And again, thank you for your attention, and I'll turn it over to Ivan to tell you about his neat results.
Thank you so much, Dr. Wright. It sounds like you have much more to say on this subject. We could hear you for a few more hours, but thank you so much for clarifying some of those subjects for her. I'm sure people will have some questions for you at the end. So yeah, now we're going to turn it over to our co-founder at Solaris Bio, Dr. Ivan Babak.
Thank you. Thank you, Dennis, for that talk. Let's see if I can advance slides here. Yeah, so one of the things that we at Solaris Bio really appreciate the work that Dennis did is early on in their drug discovery program, they wanted to make sure that any of the changes that they were making to this molecule or any of the scaffolds that they were putting together, they wanted to make sure that they were not disrupting the binding of that molecule to its intended target. So they want to correlate the IC50s that they were getting early on, that they were excited about, with the fact that it's actually binding to its intended target, not some secondary off-target effect. And so they incorporated cell target engagement early on in the process that actually helped drive this program forward. And so some of the things that drug discovery programs can incorporate when they look at target engagement is either a biophysical assay or a cell-based assay. So biophysical assays are really great and have been around for a while, but they're only good with so-called easy targets. And easy targets are targets that can be expressed outside of their physiological environment. So they can be bacterially expressed as recombinant proteins, and then they can be used in artificial environments. So for example, SPR, immobilizing a recombinant protein onto an SPR chip and then flowing over potential ligands. So it works really well for those kind of targets. But the majority of therapeutic targets out there are so-called challenging targets. And these are targets that need to exist within the physiological environment of the cell. So trying to express these as recombinant proteins is a real challenge. So this is where cell-based target engagement assays come in. Here at Solaris Bio, we have developed what's called a microtag cell target engagement technology. And in the case of DHFR, this is really what the technology looks like and how it's incorporated. So the technology is based on enzyme complementation. First of all, you would tag the protein target of interest with the 15 amino acid tag, express that in a cell line of interest. So this now becomes your microtag reporter cell. Treat the cells with the intended ligands, and then apply a thermal challenge. So with the thermal challenge, what's happening is you're forcing the denaturation of that particular target. And when it denatures, the hydrophobic amino acids get exposed, and then you start to get an aggregation happening. And so now you start to get a melting and an aggregates being formed. So you're losing that 15 amino acid tag within the aggregates. We add detection reagents after applying that thermal challenge, and the detection reagents here are a tag binding protein. So the tag binding protein would associate with that 15 amino acid tag, and we have a fret substrate that has a donor and a quencher fluorophore. They're really close together, and cleavage of that substrate by this tag binding protein and that 15 amino acid microtag enzyme complementation will cleave that fret substrate, releasing separating the donor and quencher, allowing a fluorescent signal. Now, what's unique to our technology is not only can you use this in conventional plate readers, but because it's a fluorescent signal that's actually generated through an enzyme event, so an enzyme-driven increase in fluorescent signal, we can use real-time systems such as qPCR machines or live cell imagers. So this is a really unique technology now that can advance cell target engagement beyond just static readouts with the plate reader. Just some of the highlights: it works by enzyme complementation, so that microtag associates with a tag binding protein to create an active enzyme. We interrogate the ligand binding to its target through a thermal challenge. So we use thermal shift assay, and our signal is a fluorescence readout. So it's the first fluorescence readout for cell target engagement strategies. The technology is target agnostic, so it works great with these so-called easy classical targets, for example, MTH1, but it also works with more challenging targets. And we have case studies for transmembrane proteins such as EGFR. We have case studies for GPCRs, as well as for other challenging targets that are nuclear localized, for example, transcription factors such as c-Myc. So we have some nice case studies for c-Myc. We have case studies as well for enzymatic targets for example, KRAS and of course, DHFR. So for the DHFR target engagement study, when we onboard this particular project, it starts with, first of all, a quality control, where we want to make sure that that tag that we're adding to the protein target of interest is not interfering with the target in any way with respect to the expression of that target. We want to make sure that there's no significant changes, such as proteolysis of the target, that are as a result of adding the tag. So the immunoblot in panel A using a microtag antibody demonstrates that the DHFR tag with the 15 amino acid microtag runs at the expected molecular weight. There's not significant degradation. So we know that the fluorescent signal is going to be coming from that main band that you see on the immunoblot. In panel B, this is a test to make sure that that microtag now is amenable to enzyme complementation and works in our assay system. We can get fluorescence generated. And then panel C is just a thermal profile of the DHFR microtag construct. So applying a heat gradient, we can identify a temperature at which 50% of the protein is melted, and that's about 41 degrees for DHFR in this case. And that's the temperature at which we were now interrogating the ligand binding. So in this study, we overexpressed the DHFR alpha microtag construct in HEC293 cells, incubated the cells with either 1162 or methotrexate, and then added our detection reagents, and we could see the fluorescent signal that was generated by 1162 gave a much higher fluorescence. And also the EC50 of target engagement was significantly better than methotrexate, so about 10 times better. So 1162 had an EC50 of target engagement of about 4 nanomolar, and with methotrexate in this particular assay, it was about 41 nanomolar. So this was just confirming that 1162 and methotrexate both bind to DHFR. So 1162, the structure of that molecule did not diverge significantly from methotrexate that was now binding some other proteins. It was still able to bind to its intended target, DHFR. So at Solaris Bio, we have not just this technology, but we actually have a target engagement platform. So we're very unique in this space in that by platform, I mean, we can go end to end, discover novel chemical matter in the beginning of your drug discovery program. In the middle, we can validate all the hits coming from this discovery program, validate these for binding directly to your target of interest. And downstream of the drug discovery program, we have a real-time analysis that we're actually pretty happy about and have some case studies that we're developing. So for example, live cell imaging is one of the real-time cell target engagements that we're developing. And only we can do this now because it's our chemistry allows us to do this. So one of the main features of what we've been working on and developing has been in partnership with Thermo Fisher Scientific. And this is where we're using now the real-time target engagement strategy. So this is where we get to use qPCR machines that are highly sensitive and have the ability to program various heating steps. So this has a unique feature in that it streamlines the whole process of cell target engagement. So thermal shift assays rely on first of all, identifying a temperature of melting or a temperature of aggregation for an intended target. With this novel real-time cell target engagement strategy, we abrogate the necessity for identifying a melting temperature, right? So now what you can do is just overexpress your target in a cell line of interest. 24 or 48 hours later, you can take those cells, put them into a PCR plate, add your inhibitor or ligand of interest or pools or number of compounds that you want to screen, along with the detection reagents, and immediately put it into a qPCR machine. And here with the qPCR machine, what we do is we incorporate steps of heating. So rather than taking a thermal cycler and every single well in the thermal cycler, you'll use a heat gradient and then transferring those to a conventional plate reader. That one particular well with the cells, with the inhibitor, is now going to be interrogated at multiple temperatures. So we start the step gradient, for example, in this case, you're looking at the graph on the right. We start interrogating at 40 degrees for a few minutes, then we detect the signal, and then we increase the temperature next by 5 degrees, now to 45 degrees, detect, and then keep increasing in increments of 5 degrees up until 70. So what you can see on the DMSO control, which is the red line, you start to see a plateauing of the fluorescent signal. So as you increase the temperature, that target is now denaturing, unfolding, aggregating, and so that microtag is no longer able to form an active enzyme, take part in enzyme complementation with the detection reagents. Whereas the test compound, which is the black line, you start to see the separation from the DMSO control at about 45 degrees. So that's the point at which now this target starts to unfold, but the presence of that test compound is going to stabilize it from being denatured. And so we get the separation between the DMSO control and the test compound. That separation between the two lines is how we can quantify the amount of stabilization, so the amount that of binding for that particular ligand and target interaction. So we're really excited about this partnership with Thermo Fisher coming out on their website soon. Here's a case study for using that real-time target engagement strategy. Again, the quality controls first to make sure that the tag that we've introduced is not impacting the expression and the folding, I guess, of the protein. So this is a microtag antibody immunoblot showing that expressed at the correct molecular weight, and there are no proteolytic fragments. So the fluorescent signal is coming from that one particular overexpressed protein. The tag itself is amenable to enzyme complementation, generates significant fluorescence. So now it's ready for testing with any drugs, inhibitors, or activators. So in this case, we were using an AstraZeneca compound. So it's a well-characterized, commercially available compound that's been described as a MAPK1 binder. As a negative control, we have DMSO as a negative control, along with crizotinib, which is not expected to bind MAP kinase. And we performed the step gradient using the qPCR machine, as mentioned previously. And so what we can see is halfway up at about 55 degrees, we start to get the separation between the AZD compound and the DMSO and the negative control compound. So you start to get the separation of the signals. And what's interesting here is MAP kinase. If you do a thermal profile to identify melting temperature for MAP kinase, it's actually 54 degrees. So we start to see here at about 55 degrees Celsius, we start to see the separation of the signal, indicative of a binding event, of a stabilization of that target in the heat challenge. In panel B, this is how we quantify the amount of stabilization, and that's by area under the curve. So we determined the area under the curve for DMSO, the negative control, and the AZD compound, subtract out the DMSO area under the curve, and that's the bar graph that you see in panel B. So the amount of stabilization of the AZD compound is significant, and the amount of stabilization of the negative control crizotinib is actually flatline at 0. Panel C and D, these are dose response of the AZD compound, which is expected to bind, and the crizotinib compound, which is not expected to bind. So the AZD compound with a dose response, you can see at about 3 nanomolar, it starts to separate the signal. So now you start to get stabilization of the target as you go above 3 nanomolar. And what we can do with this now is you take the area under the curves for these, subtract out the DMSO area under the curve, and then we can plot that on the y-axis along with the inhibitor concentration to identify what we're calling it's an in-cell KD measure, and a measure of the target engagement affinity here. And so we call it an in-cell KD just because we're adding the cells, the inhibitors, and the detection reagents together into a pool prior to any sort of equilibrium that can take place between the target and the ligand. So for AZD, in this particular case, the AZD 0364, we have a in-cell KD measure of about 4.3 nanomolar. So we're really excited here at Solaris Bio. There's a lot of new things coming, so please stay tuned for some of our future webinars. And I'll hand it over.
Thank you so much, Dr. Babic. That was excellent, very exciting technology. So that was the end of our presentation, but we do have time for a few questions. So if anything came up during the presentations, please go ahead and pop them either in the chat or the Q&A. And while you're looking at these exciting future webinars, I'd also love it if you could answer a question for us. A poll will pop up on your screen, and if you don't mind just answering that question. All right, while those are coming in, we'll bring in our speakers again. And we have a question here. Did you guys use a negative control? And if so, was there any non-specific binding or noise from that profile versus compounds used?
I'm not sure if that was meant for the target engagement study or if it was meant for some of the other studies that Dennis did. But for target engagement studies, we typically incorporate negative controls, for example, actin or GAPDH. So we can incorporate that as part of the studies. Now, I'm not sure if we did that for this particular study. In other cases, we also have, for example, KRAS mutants. We test both wild type and the mutant forms. And that we have other cases of other types of enzymes where as a negative control, we can mutate one of the amino acids and test that alongside the actual wild type protein or some other mutant version of the protein. But that's a good question, and we typically incorporate these types of negative controls in these assays. And the noise is not really that much, so we don't typically see binding, because a lot of these are very specific molecules. They're not sticky molecules. So we've worked with sticky molecules that tend to be very promiscuous and bind to a lot of different things. Yeah, I hope that answers the question.
Yeah, thank you. Perfect. All right. Dennis, could we go to you? Could you tell us about the next phases in your drug discovery program for DHFR?
Yeah, so so essentially we've got a couple different we work in a few different areas. And our two biggest areas are in antifungal DHFR inhibitors. Obviously, I didn't talk about those today, but again, the design principles are very similar. Again, where we're looking at resonance time as a driver of anti-proliferation. And for this particular project, we're now doing larger scale animal studies, efficacy studies. I think I showed you some of the data from that, and we're doing that with some additional AML lines and some PDX models. So we're kind of gearing up to do a lot of animal work right now on that before we hit our next phase of that.
Okay, exciting things to come then. Perfect.
We hope, yes.
Dr. Yes. Dr. Babbitt, could you tell us what makes a drug target challenging?
Sure. So some of the challenging targets that we've worked with, for example, are transcription factors. So transcription factors, if you try to express those outside of the cell, so bacterial express and develop recombinant proteins so that you can work with them in vitro, it just doesn't work. And so these are the typical challenging targets that really cannot be outside of their physiological environment. So this is why it's important to incorporate a cell target engagement assay where you utilize intact cells for some of these studies.
Okay, perfect. And then I think we have a follow-up from the first question. As a clarification, is your detection molecule cell permeable, or is there a cell lysis step prior to detection?
So with these, when we use intact cells, we can do a pre-incubation step just with the cells. And then when we have to add the detection reagents, we add a non-denaturing lysis step that is gentle detergent that pokes holes into the membrane, allowing the detection reagents to get in. So the detection reagents are not cell permeable, but the way we get them in is through a gentle lysis.
Okay, perfect. All right, and then we have one more for Dennis. Could you speak to the relative cellular concentrations of methotrexate versus 1168?
So usually, in almost all cell assays, we find that 1162 is about 10-fold more active than methotrexate. So that's been pretty consistent. That's been consistent both for activity against proliferation of cancer lines, as well as we looked at some markers like production of inflammatory cytokines. So we find it's about an order of magnitude sort of better, sort of across the board. And if you kind of look at what's really nice, if you look at Ivan's data, we see that about an order of magnitude difference in the engagement in the cell. So, we think that I think is really powerful about this is that we really can see that correlation with their assay of longer and more, longer duration inhibition, more stabilization of the enzyme in the cell. And then that correlates almost like, oh, wow, we were kind of surprised how well correlated to the difference in concentration, say EC50 values.
Fantastic. Thank you both so much for answering those questions. It looks like that's everything for us today. So yeah, thank you both for giving us such clear information. If you would like to contact us, please reach out at hi@solarisbio.com. We also have a website, solarisbio.com. Stay tuned for upcoming webinars and have a great rest of your day, evening, or tomorrow. Bye, everyone.